Today we're going to be looking at Chapter 3, Part A. Here's an overview of the chapter. So we'll start with cellular units, which are the smallest or basic unit of life. Then we'll look at what's inside cells, and of course we'll delve deeper into that. Let's start with Robert Hooke, who was the first to observe a plant cell with a crude microscope in about 1665. So he had a microscope that had enough magnification to observe the basic shape of separate plant cells, and this was from a slice of a cork. And he noticed that the tiny compartments, which are now called cells, appeared to resemble the compartments monks would live in. And monks lived in rooms called cells, so that's why he called what he observed a cell, and it stuck. There's also Rudolf Virchow, which is a German pathologist, and he had this idea that cells arise from other cells, which seems like a simple idea. It doesn't seem like anything that stands out now. But back in his time, so 1600s, early 1700s, there was the idea of spontaneous generation, which is a widely accepted idea. This idea existed for 1900 years and was even supported by experiments or reasoning. But Rudolf Virchow challenged this widely accepted theory, and he proposed that cells come from pre-existing cells, or that units of life come from pre-existing units of life, whereas the spontaneous generation theory stated that things that are living could just arise from non-living things, like fleas would arise from dust, or rats from straw, or even maggots from flesh, like rotting meat. So ever since this kind of early research into the cellular basis of life, so that's about early 1600s all the way into the late 1800s, cell research has just exploded. There's been a lot of fruitful and very key concepts that have been developed, and they've been put together in the cell theory. So again, these key concepts that we're about to look at are known collectively as the cell theory. So here are the 4 tenets of the cell theory. Some textbooks will add another, or take away some, or combine the way they're worded, but these are the basic tenets. And first and foremost, the cell is the basic structural and functional unit of life. So if we were to define the cell properties, or if you were to define the properties of life, you would find that cells are going to fit all those properties of life, and that all the properties of life fit what cells do. We also know that continuity of life has a cellular basis. That's the same idea we were talking about with Rudolf Virchow, that all living things are made of cells and come from pre-existing cells. So unicellular and multicellular organisms arise from pre-existing cells. And then last but not least, what will become a major focus in A&P, especially as we move next into histology, is the fact that cells look different. They differ in size, shape, the subcellular components, and functions. So while most cells do have a nucleus, a cell membrane, and cytoplasm, the variety of organelles they have within, or how much of a particular organelle, is really going to match their function. And so that just brings back from Chapter 1 the complementarity of structure and function, that tenet that is going to hold true throughout anatomy and physiology. So how well the entire organism functions is really going to depend on individual and combined activities of all of its cells. So again, if you don't know what I'm saying when I say complementary structure and function tenet, go back to Chapter 1. So here's just an idea at the breadth of cell diversity. So there's over 200 different types of human cells. You find some in your bloodstream, so within blood vessels. There's even more cells that make up your blood vessels, like epithelial cells and connective tissue cells. There's fat cells, which is a type of connective tissue. So there's going to be broad general categories that we fit all types of cells within. Those broad general categories are things like muscle cell, or epithelial cell, or nervous system cell, like the neurons and neuroglia you find in your central nervous system. And then, last but not least, is going to be connective tissue. Connective tissue is by far the broadest category. It includes things like these fibroblasts up here, the fat cell, as well as many different types of cells that make up things like tendons in your joints, even the cartilage in your ears and your nose, or in your ribs. But let's take a step back before we get there. Again, a lot of that detail will come in Chapter 4, which is looking at histology, or the study of tissues. And tissues are made up of cells, so we have to start with a basic idea before we build the tissues. A generalized cell is made up of these three things: a plasma membrane, cytoplasm, and a nucleus. The nucleus is genetic material containing. It's usually thought of as the control center. It's in the center of a cell. Then surrounding that, but within the plasma membrane, is the cytoplasm. Cytoplasm is made up of cytosol, which is a gel-like fluid, and organelles, which are cell machinery. When you put those two together, you have cytoplasm. And then last but not least, acting as a boundary to essentially give the cell an inside and an outside, you have the plasma membrane. And regardless of the type of cell, all cells are going to be composed chiefly of carbon, hydrogen, oxygen, and nitrogen, and some other trace elements like phosphorus and sulfur. These components that we see in many biological compounds, and biological compounds make up cells. Here is a basic animal cell talking about humans and anatomy and physiology, and so this is a cell that you might see in the human body. It has a nucleus, so we would actually call this type of cell a eukaryotic cell, as opposed to cells that don't have a nucleus, which we'd call prokaryotic cells. For the rest of anatomy and physiology, I won't mention prokaryotic cells, and it's safe to assume that anytime I mention a cell, we're talking about a eukaryotic cell with a true nucleus. We can see that the nucleus is in the center. It contains a nucleolus, which is an area that creates rRNA. I won't go into a lot of detail yet, but then we have a nuclear envelope that has pores, or nuclear pores. That's so that mRNA or RNA can get in and out of the nucleus. Usually closest to the nucleus is this sac-like blue structure with the dots, the red dots, which are ribosomes, and the blue structure that is dotted is known as the rough endoplasmic reticulum. There's also free ribosomes. We can see some of those floating around in the cytosol. There's also a tubular structure near the rough endoplasmic reticulum. That's the smooth endoplasmic reticulum. You also have things like mitochondria, which create ATP, usually depicted in green, and further away from the nucleus than the rough ER, or rough endoplasmic reticulum, is the Golgi apparatus, which is kind of like the post office for the cell. It can create things called lysosomes. There's also peroxisomes. Lysosomes tend to have digesting functions, whereas peroxisomes tend to have detoxifying functions. And if we're not digesting or detoxifying, sometimes these bubbles, which contain substances, are just called vesicles, and they're transport bubbles, which are then called transport vesicles. There's also a pair of centrioles. Centrioles produce microtubules, which can help form things like the spindle apparatus for when a cell divides. And then that's the basic stuff that we see here. We'll talk more about chromatin, other things like types of secretion, intermediate filaments, which is part of a cytoskeleton that gives the cell shape. But one of the first things we'll talk about will be that plasma membrane, this lipid bilayer that covers the entire cell, and again acts as a boundary that gives the cell an inside and an outside. So let's look at that plasma membrane. So first and foremost, it's a barrier. Very simple. It gives the cell an intracellular environment, which contains intracellular fluids, and an extracellular environment, which contains extracellular fluids. So ECF is an abbreviation for extracellular fluids, and that's just substances found outside the cells, whereas ICF is an abbreviation for intracellular fluid, and those are just substances found inside cells. The plasma membrane does play a dynamic role in cellular activities, and what that means is it's selectively permeable. It gets to decide what comes in and what leaves the cell. So that control gives it a dynamic and very important role in cellular activities. You may hear the cell membrane instead of plasma membrane. You may also hear lipid bilayer or phospholipid bilayer as words used to describe that plasma membrane. So let's just take a minute though and talk about what could be outside the cell. Well, it could be interstitial fluid, which is a fluid that tends to bathe tissues or the outside of cells, because we know tissues are made up of cells. So cells are generally submerged or bathed in that type of fluid. There's also blood plasma, which is majority water, and you'd find things like red blood cells, white blood cells, and platelets surrounded by this blood plasma. It's also within blood vessels, so it would be within blood vessels coating the inner lining of epithelial cells of those blood vessels. There's even cerebrospinal fluid, which is fluid surrounding nervous system organs. So it's not a neuroglial cell, it's not a neuron, it's a fluid surrounding those structures. You would specifically find cerebrospinal fluid in the central nervous system, so around the brain or the spinal cord. There's also cellular secretions that may not always be extracellular, but once secreted from a cell, they are, such as saliva, mucus. Some cells are unicellular and secrete mucus like goblet cells. There is also the extracellular matrix, because outside cells it's not just fluid. So body fluids like blood plasma, interstitial fluid, and cerebrospinal fluid may be intermixed with other cytoskeletal elements, or things that are protein-based, things that can act as a glue to hold cells together. Think of it kind of as fibers weave throughout a gel-like fluid. The structure of the plasma membrane is very dynamic, very complex, but simple to describe. We could do a whole course on the cell membrane and how it allows things in and out, but we just want to kind of get the bare bones of its structure and then later its function. So it does consist of lipids. Because these lipids make up a membrane, they're sometimes called membrane lipids. Because there is going to be two layers of these lipids, it's oftentimes called the lipid bilayer. And this type of lipid that makes up the plasma membrane is a phospholipid. So sometimes instead of just lipid bilayer, you may hear phospholipid bilayer. And bilayer is just referring to the two layers of lipids that help form the structure of the plasma membrane. So there are also specialized membrane proteins that are embedded in this lipid bilayer. They float, they're not stuck in any one location. So oftentimes the plasma membrane in structure is referred to as a fluid mosaic, or as following the fluid mosaic model, meaning that just because a membrane protein is located in one location at a certain time doesn't mean it can't move through the lipid bilayer, through those membrane lipids, and relocate, but it'll still be found in or embedded in that lipid bilayer. If you were to look in a light microscope at a cell, you may be able to see a fuzzy layer on the outside of the cell. That fuzzy layer is basically a poor rendering of the glycocalyx, or the surface sugars. Think of the glycocalyx as surface markers that identify cells as either supposed to be in your body or not supposed to be in your body, but it also identifies them as epithelial cells or muscle cells, or however type of cell they need to be identified. Maybe they're an immune cell. And then membrane structures that help hold cells together are cell junctions. And we'll look at different types of cell junctions. Some are communicating, some act as a molecular zipper but allow some stretch, and others are impermeable and don't really allow stretch or tearing. So here's that fluid mosaic model. We have, point them out, we have 1 and 2 layers of lipids. You can see that the phospholipids have a head and 2 tails. So they have a head that contains a phosphorus and 2 fatty acid tails which contain carbon and hydrogen. And because these lipids are rather smaller, there's only 2 fatty acid tails, the cell membrane is relatively thin. But we also have these embedded proteins. The ones that are embedded all the way through are called integral proteins. The ones that are attached to the integral proteins but are only found on one side of the cell membrane are called peripheral proteins. So you can think of these integral and peripheral proteins as at least partially embedded or plugged into the cell membrane. They're dispersed throughout that cell membrane, and they can float through these plasma membrane lipids. So they're not going to necessarily be stuck in that one location, but they will remain in the plasma membrane. And we can even see some of these carbohydrate sugar molecules that make up the glycocalyx, and will look like a fuzzy layer on the surface of the cell if we used a light microscope. There's even a fluid, which is extracellular fluid, that contains proteins and fibers making up that extracellular matrix. So the lipid bilayer with its protein molecules dispersed in it creates that fluid mosaic model. We saw that it consists mostly of phospholipids, but also contain things like glycolipids. So when we have sugars attached to phospholipids, that makes a glycolipid. What you might not have noticed were cholesterol molecules. Cholesterol molecules can change the fluidity of the cell membrane. So adding more or less of them will change the fluidity of that fluid mosaic model. Again, glycolipids are just lipids with bound sugar groups. Phospholipids have a polar head, that's where the phosphorus is, and nonpolar fatty acid tails. We would say that since the head is polar, it's like water, so it's water-loving or hydrophilic, whereas the nonpolar tail wouldn't mix with water, so it's closer to oil, just like fatty acids are oftentimes considered lipid-like or parts of lipids, so the tails are going to be hydrophobic. So that means that phospholipids, which make up most of the plasma membrane, are going to have water-loving and water-fearing areas, or hydrophilic and hydrophobic areas. Any type of molecule that has both areas, an area that likes water and an area that does not, we call that type of molecule amphipathic. If it just liked one or the other, we would just call it hydrophilic or hydrophobic, but when both are present, we call that type of molecule amphipathic. So let's look at phospholipid structure closer. Now imagine this bilayer going all the way around a cell. We've just taken a small piece, and what we're able to do is see that the tails are going to be located in the middle of the lipid bilayer, and the outsides are going to be located either close to the extracellular space or close to the lumen of the cell. So the heads are going to be directed towards areas that are watery. So there's a lot of water in the extracellular fluid. There's a lot of water in the intracellular fluid. And the nonpolar tails don't like water, so they're hiding inside the lipid bilayer, whereas the heads are hydrophilic, so they can face the watery environments. So hydrophilic head, hydrophobic tail, it's an amphipathic molecule made up of fatty acids, glycerol, and a phosphate group. And if you need to go back and look at the structure of a phospholipid, go back to your textbook and look at chapter 2. So let's talk about the composition of the plasma membrane. We know there's a lot of phospholipids, but how many? It's actually about 75% made up of phospholipids, hence the other name being the lipid bilayer. It's 5% glycolipids, which help make up that glycocalyx, which are surface molecules, and then 20% cholesterol, and again cholesterol increases membrane stability, but decreases fluidity. So a little bit less cholesterol, a little more fluidity, a little less membrane stability. The functions you can probably guess at. 2 we've already suggested. It's a barrier. It gives the cell an inside and an outside, so it's a simple mechanical or physical barrier. It also gets to decide what comes in the cell and what leaves, so it's selectively permeable. It also is able to generate an electrochemical gradient. When we get to chapter 9 or talk about skeletal muscle and then later the nervous system, we'll really delve into this electrochemical gradient. We'll see how certain cells, like muscle cells and neurons, are able to generate and maintain electrochemical gradients that are required for their function. The plasma membrane is also a communicating area. Those surface markers do things more than just kind of sitting and acting as a marker for saying what the cell is. Then there's cell signaling. So we'll look briefly at second messenger systems and how that would work, how we would get a signal from outside the cell to inside the cell without something actually entering the cell, which is called signal transduction. So next let's move on and look at the proteins that are embedded in the plasma membrane. So you have integral proteins, which are firmly inserted all the way through the membrane. If it goes all the way through from one end to the other, not just partially within the membrane, it's actually called transmembrane. These proteins generally also have hydrophobic and hydrophilic regions. That's why parts of the protein are able to hang out near the hydrophilic heads of phospholipids, and the other parts are able to hang out near the hydrophobic tails of the phospholipids. There's different types of integral proteins, some that transport, some that act as enzymes, and others that are still going to be receptors. Receptors for signal transduction function really well with amino acid hormones of the endocrine system. There's still also going to be parts of those hormone systems that need enzymes. And then transport proteins are the ones we'll focus the most on, and there can be channels and carriers that act as transport vessels. Then there's peripheral proteins, which are generally attached to an integral protein, but are on the periphery, so they're either outside or inside the cell. Sometimes these peripheral proteins will have glycolipids on them, and they'll act as cell-to-cell recognition molecules. They also have enzymatic activity and some motor protein activity, but cell-to-cell links, support, that's going to be what we look at mostly for our peripheral proteins. And next we're going to talk about cell junctions, because some of these proteins we're looking at function in forming cell junctions. Because even though some cells are free, like spermatozoa, or blood cells, like your red blood cells, and they're floating either in reproductive organs or inside your blood vessels. Not all cells are free. A lot of them are bound to other cells that look like them or have a similar function, or maybe they're a part of a tissue that's helping to make up an organ. So again, if we go back all the way to chapter 1 and look at that organization of the human body, we can see that most cells are actually bound together to form tissues and organs. Here are the 3 types of cell-to-cell junctions, or cell junctions, we can just call them membrane junctions, that we want to know about. And these are the most important in securing cells together. So 3 types. There's a tight junction, a desmosome, and a gap junction. So that's our list, but let's go look at each one in more detail. So first there's tight junctions. Tight junctions are impermeable, meaning wherever they're located in that area where the cells are being held together, you wouldn't be able to get anything through. Now, a tight junction isn't always going to be found all the way through a cell connection to another cell, but wherever they're located, the little proteins that kind of zigzag embed themselves in the 2 plasma membranes are going to be impermeable. We're not getting stuff through them. No solutes, sometimes not even water. Desmosomes, on the other hand, are kind of like molecular zippers. You have these 2 linker proteins. So you have these kind of plaques that have linker cadherins, and the linker cadherins link together, and it allows for a little bit of stretch. For example, if this plasma membrane pulled a little bit to the left, and this plasma membrane pulled a little bit to the right, we would just not have these cadherins linked as much. They would just kind of slide past each other a little bit, but as long as it's not something that's shearing or tearing something in half, it shouldn't completely dislodge this molecular zipper. So desmosomes are really helpful in things like your integument or your skin. They're great in your heart, so between cardiac muscle cells, because they reduce the chances of tearing when a tissue, so tissue made up of cells, is subjected to pulling forces. So again, skin and heart muscles, that's where you're going to find these types of cells. One of the reasons if you have an itch on your arm or somewhere else and you scratch it, that your skin doesn't just peel off, is because it's allowed some stretch. It's allowed some pulling force without having to tear or break down. So we should be really helpful, or really thankful for our desmosomes because they're really helpful. And then last but not least, we have our gap junctions. And you may have suspected, based on the name, that our gap junction have a gap in between them. They're made up of a ring of proteins that embed themselves between the plasma membranes of 2 linked or adjacent cells, and really small things can go through these plasma membranes. So small things like ions and water. I wouldn't send something like glucose, which is a molecule made up of multiple atoms, all the way through these 2 membranes, but small things like singular atoms or charged ions can get through. So you should know gap junctions as your communicating junction. So here's some of that information if you do need to come back to it. So tight junctions are impermeable. Their integral protein molecules were fused, not getting anything through. Desmosomes had those anchoring cadherins, and they were linking adjacent cells, but they allow some stretch. So we have our molecular zippers here. We have some give between cells, and we saw these in the skin and in cardiac muscle. And then last but not least, we had gap junctions. Again, communicating junction, because they have a gap so things can get through, but really small sugars, ions, and we're not going to send anything large through. And the transmembrane proteins that function in gap junctions were called connexins. Even electrical signals can use gap junctions, and we see this occur also in cardiac muscle. Next, we're going to move on and look at membrane transport. So we'll look at passive and active transport. And the biggest difference we're going to focus on is that passive transport does not require energy. Active transport does require energy. So because the plasma membrane is selectively permeable, it gets to decide what comes in and what leaves. Basically, some molecules can easily pass through and others do not. Because this can occur, we have to have more rules that govern how things move. So first and foremost, the plasma membrane is selectively permeable. It gets to decide what comes in and what doesn't. Because of these functions, there's rules that we have to look at that decides what can come in and what can't when certain things are present. For example, at first we could just look at what is a substance. If it's lipid soluble, it can go straight through the plasma membrane. Or even if it's not lipid soluble, but there's a channel or a carrier protein that'll move it across the membrane, then it can enter or leave, depending which way it's going. But if something is going to be hydrophilic or not lipid soluble, or if it's very large and it doesn't have a channel or carrier, it's going to be hard to get it through the membrane, whether it's leaving or coming in. So let's start with passive transport. So here we just have an experiment where you have a beaker of water and 1 drop of purple dye is being added to the water. And then nothing else is being done. We're just waiting. And over a period of time, without stirring, without heating the water, the dye molecules will spread out. They'll move from an area of high concentration of dye molecules outward to the lower areas of concentration for dye molecules. And this is a passive process. We didn't stir or add heat, so we added no energy to this reaction, but we still got movement of the dye molecules from high to low concentration. And as long as we wait long enough, eventually we will have the dye molecules spread out through the beaker. So this is not an active process which would have required energy, such as us heating or stirring this reaction. So we're going to look at types of passive transport. Another basic example before we go forward would be spray perfume in one area of a room, or spray Febreze in one area of a room. And without wafting it, without walking around the room and spraying more, eventually those odor molecules will travel across the room from the high area of concentration, which is where you sprayed them, to the lower areas of concentration. The types of passive transport we're about to look at are simple diffusion, facilitated diffusion, and osmosis. Now, passive transport we know requires no energy. Diffusion, as an example, is going to be one of the simpler types of passive transport. But if it's a type of passive transport, it requires no energy. And we call it simple diffusion because it is simple. Usually things that have a carrier or a channel can go through the membrane. Or if they fit through the membrane, or if they're small, or if they can dissolve in lipids, they can go straight through the plasma membrane. Now, there's different types of diffusion. Simple is when something does just go straight through the membrane. And then you have facilitated diffusion, which is a type of diffusion where it can go through the membrane, but only with help, only with facilitation, such as a carrier or channel protein. And then we'll look at osmosis, which is the movement of water. And as long as it's a single water molecule, it can move straight through the plasma membrane, even though it's going to be hydrophilic because it is water. If it's small enough, it can squeeze through that fluid mosaic model. There's also things called aquaporins, though, that help move a lot of water at once in and out of a cell. What we're not going to pay a lot of attention to in this chapter, but you'll talk about later in Anatomy and Physiology, and especially in Anatomy and Physiology 2, is filtration, which is also a type of transport, and that usually occurs across capillary walls, and that's usually because of pressure gradients. Whereas we're concerned with passive and active, we're concerned with our concentration gradients. So pressure gradients can also play a role. So let's look at the different types of diffusion. We have simple diffusion, and again, it's simple. So we have a high concentration of lipid-soluble molecules on the left, and they can go into the cell, so they can go left to right, because they can travel without energy from high concentration to low concentration. The reason I mention them being lipid-soluble, or even I could say they're just small, is because things that are lipid-soluble are small, or if we were to look at things like facilitated diffusion later on, have a carrier, can go in or out of the membrane. So for simple diffusion, which is a type of passive transport, we're not going to use ATP, because ATP is cellular energy, and we don't need that for passive diffusion or passive types of transport. Simple diffusion means something is lipid soluble or small, so things like oxygen, carbon dioxide, anything lipid-like, and even small molecules or single molecules of water can travel from high concentrations to low concentrations, or areas where there is a high concentration to areas where there is a low concentration. So simple diffusion just relies on the collisions between molecules in an area where there's a high concentration, and those collisions would occur more likely in an area where there is more of a substance, and those collisions are more likely to cause those molecules to scatter and move to areas with less concentration. So molecules do have a natural drive to diffuse down their concentration gradient, and I'm going to focus on that phrasing for just a second. If a molecule moves down its concentration gradient, that means it's moving from high to low. So you may have a question that says, you have a molecule that is lipid soluble and it moves down its concentration gradient. Is this an example of active or passive transport? Even though they're not saying there's energy, and maybe you don't want to assume that there's no energy, you want something else to give you a for sure hint that it's definitely a type of diffusion or a type of passive transport. The term down its concentration gradient would be your clue, because if something's going from high to low, it's passive transport, and no energy is mentioned. So this is an example of passive transport, specifically simple diffusion. And just because it's simple diffusion doesn't mean that something has to go inside the cell. It can move from an intracellular to an extracellular area as well. So here's an example that we see in your book. We have a lot of lipid-soluble solutes, or things that dissolve in a solvent outside the cell, and when they collide, they push themselves to another area. They can push themselves through the plasma membrane because they dissolve in lipids, and most of the inside of our plasma membrane is lipid-like. So they go straight through to an area of lower concentration. No energy required. This is simple diffusion. Now there are things that can affect diffusion rate: temperature, particle size, concentration gradient. We tend to see that diffusion rate increases if you increase concentration gradient. If the area with the highest concentration was to have an increase in its concentration, then things would move from high to low concentration even faster, because those particles would collide in more abundance. If you increase temperature, particles tend to make more collisions, because if you increase temperature, particles tend to gain energy and move faster. So the warmer something is, the faster the reaction. The same thing goes for particle size. If something's smaller, it can move faster, which is going to increase the collisions between molecules in the high concentration area and cause them to move to the low concentration area. Next, let's look at facilitated diffusion. Now, there are certain molecules that can't just go straight through the plasma membrane because they're hydrophobic. They're more like water, or they're large, like glucose, amino acids, or charged ions. And so things like that, since they can't just go straight through our lipid bilayer, they have to be transported. They need help. They need facilitation. So that's what we have facilitated diffusion for. So facilitated diffusion is going to be the movement of hydrophobic molecules or large and charged molecules through a carrier channel protein. So this is oftentimes called carrier-mediated or channel-mediated facilitated diffusion. So let's look at an example. You can have, first and foremost, a channel protein, which tends to be always open, and then a carrier protein, which tends to change shape or change conformation. We're still talking about passive transport. We just made it a little more complicated because these things need help to get through the membrane. But we're still moving things from a high concentration to a low concentration, just with the help of a carrier or channel protein. So still no ATP is being expended, but things like water, charged molecules, and large molecules would need help from a protein. And that's what we have facilitated diffusion for. So here is an image from your textbook. We can see lipid-insoluble solutes, so things that are hydrophilic, things like sugars, or maybe it's an ion. They're using a carrier protein to move them from an area of high concentration to an area of low concentration. And that just happens to be from outside to inside the cell. And these carrier proteins tend to be specific. So for example, here we have a carrier that is specific for glucose. So it would only carry glucose molecules, whereas channel proteins tend to not be specific, and they'll carry anything that fits through their channel. So there tends to be a little bit of enzymatic activity, a binding of a substrate to a protein in order to cause the shape change for carrier proteins, and that would allow the molecule to process from one side of a cell membrane to the other. So again, our channels are not going to be specific. Leakage channels are always open. You do have some that are gated, and we see those in the nervous system. And they're usually controlled by chemical or electrical signals. Some of these channels are actually called aquaporins, and they're going to be channels that are going to be able to move water, sometimes ions, but if you move water through a channel, that's called osmosis. So here's an example of an aquaporin. We have the lipid bilayer. It's an integral protein embedded in that lipid bilayer, and we see an area of high concentration of water molecules and to a low concentration of water molecules. It's important to note that with simple diffusion and facilitated diffusion, we were moving solute. We're moving solute from high to low concentration with no energy expending. Whereas with osmosis, we're moving water from an area of high water concentration to an area of low water concentration without using any energy. If you like just talking about only the solute and not switching what we're moving, you could say that during osmosis, water is going to move from an area with low solute concentration to an area with high solute concentration. But I much prefer to say that all you have to know during the passive processes is what is moving. And if you focus on what is moving, then during a passive process, the things that move are always going to move from high to low concentration. So for simple diffusion, we're moving solute, so it moves from high to low concentration. For facilitated diffusion, we're moving solute, so it's going to move from high to low concentration. And for osmosis, we're moving water, so water is going to move from high to low concentration. And that movement of water, which is a solvent, not a solute, across a selectively permeable membrane, that is specifically osmosis. It's a very particular and specific passive process. And water can diffuse through the plasma membrane by itself, but more often than not, we'll use these aquaporins. So osmosis is important. Water moves by osmosis, again, from areas of low solute or high water concentration to areas of high solute concentration or low water concentration. And when osmosis occurs, the water would enter or leave a cell. And for animal cells that don't have a plant cell wall, that can be dangerous. You can cause cells to bust or to shrivel. So when osmosis occurs and water enters or leaves a cell, that means that the limits or the amount of water is also going to change. And a change in cell volume can disrupt cell function, especially in neurons. We want to watch that. So let's talk about some terms used to talk about that movement of water. So again, osmosis is the diffusion of water across a selectively permeable membrane. It usually uses a channel, like an aquaporin, but once the movement has occurred, how do we compare two solutions? So the terms I'm about to mention are used in order to compare solutions. So we have hypotonic solutions, hypertonic solutions, and isotonic solutions. Let's talk about the prefixes of those terms. Hypo means below, hyper means above or higher, and isotonic means same. So if we're comparing two solutions and they're isotonic to each other, that means the solutions have an equal amount of solute and water. If we're comparing two solutions and one solution is hypertonic compared to the other, that means the hypertonic solution has more solute, so more things dissolved in water, than the other solution. So it would have higher solute, lower water. So what you should be picking up on is the prefix is telling you information about the solute, because hypo means below or lower, and in a hypotonic solution, that's a solution that has less solute and more water than another solution. But I can't just use these terms if I'm talking about one solution. If I'm talking about a beaker of salt water, this term, hypo or hyper or iso, not useful. But if I'm talking about beaker A filled with salt water and beaker B filled with salt water, I can use these terms because maybe one of the beakers has more salt. And if one of the beakers has more salt, that beaker with a higher solute concentration is hypertonic to my other beaker. Let's say beaker A has a really high solute concentration. There's a lot of salt dissolved in that water. Beaker B has just a little bit of salt dissolved. So beaker A is going to be hypertonic to beaker B. Here we have a U-shaped test tube. And we have a selectively permeable membrane, but this membrane is impermeable to water. So the solute could move. This is impermeable to solute, so the water can move, but the solute cannot. And this solution only has a couple of dots, that's the solute. The solution on the right has a lot of dots, that's the solute. So I would say that the solution on the right is hypertonic to the solution on the left, because the solution on the right has a higher solute concentration and less water compared to the solution on the left, and vice versa. Now in this U-shaped beaker, with a membrane that's impermeable to solute, I would not expect any of the dots to change size. But the water can move. We could actually see that water will move from this side to this side because there's more water over here on the left than there is on the right, and we know water can move via osmosis. So when water moves, it moves from an area of high concentration of water to an area of low concentration of water. Well, what if you're having a hard time figuring out which side has more water? Because first and foremost, this looks pretty equal to you. Well, then just remember that water also moves from an area with less solute to an area with more solute. So that would also answer the question, which way is water going to move? And it's going to move to the right. So let's try a couple of questions. So here we have 4 different examples. We'll do these last, but we have 2 situations down here. Now 2 situations, but the information is all that's going to change because there are 3 dots on the left situation here, 6 on the right, and the same thing over here. The information that changes is the information telling us about this membrane. So on the left situation, this is a semi-permeable membrane, and it's impermeable to solute, so our dots can't move. Over here, the membrane is permeable to solute and water, so the dots and the water can move. So the first question for both situations is, does solute move through the membrane? And then if so, which direction? Well, this membrane is impermeable to solute, so the solute doesn't move. This membrane is permeable to the solute, so the solute does move. And we know solute likes to move from areas of high concentration to low concentration. So our dots are the solute. So if the solute could move in this situation, it would move from right to left, but it can't. Over here it can, so we'll actually see it move from right to left. Question 2 says, does water move through the membrane? If so, which direction? On the left situation, water does move because the membrane is only impermeable to solute. The water, however, can move. On the right, the membrane is permeable to water and solute, so the water can move too. Since the situations are the same, we're going to see water move in the same direction. We know water moves either from high to low water concentration or from an area of low to an area of high solute concentration, whichever way you want to remember it. So for the first situation, solute doesn't move, but the water moves to the right. For the next situation, solute does move, it moves to the left, and water also moves to the right. And for these situations, we just want to know, when comparing 2 solutions, is solution that is listed first hyper, hypo, or iso compared to the second solution. So let's look at the first situation. A compared to C. A has more solute than C, so A is hyper to C. C has less solute than B, so C is hypo to B. B has more solute than D, so B is hyper to D. And then A and D have the same amount of solute, so they're going to be isotonic. Now, what does this have to do with actual cells? Well, it has to do with tonicity, which is the ability of a solution to cause a cell to shrink or swell. Because isotonic solutions, hypertonic solutions, and hypotonic solutions, they'll all have an effect on our own cells if, for example, inserted into our bloodstream. So if you give somebody an isotonic solution in their bloodstream, their cells are going to retain their normal size and shape. So same solute water concentration is what isotonic means, and we're going to see that water moves in and out, but the cell retains its original shape, so it shouldn't lose function. Whereas if you give someone a hypertonic solution, then because that contains a higher concentration of solutes than are present inside the cells that are being exposed to it, the cells are going to lose water by osmosis. Cell will exit. The cell will leave the cell, which has a lower solute concentration, and move to the area of higher concentration, or to that hypertonic solution. Then vice versa for a hypotonic solution, the cell will take on water by osmosis until they become bloated and burst. That bursting is called lysing. So in a hypotonic solution that contains a lower concentration of solute than are present in cells, we'll see water enter and again cause a cell to swell, then bust. That's very dangerous. It can destroy cells, but it's just as dangerous to cause cells to shrink. And this shrinking is generally called crenation. So here's a clinical or homeostatic imbalance, just talking about what we just looked at, that intravenous solutions of different tonicities can be given to patients suffering different ailments. So even though isotonic is the most commonly given in order to increase blood volume and blood pressure quickly and not change cell shape, there are certain situations where you'd want to use hypertonic and hypotonic solutions, but most of the time you would not. So you may want to use a hypertonic solution if you're treating an edematous patient, a swollen patient that has fluid that is leaving cells, because that would pull water back into the blood. Or a hypotonic solution should not be given really any time because it could result in dangerous lysing of red and white blood cells. So some situations we would want to use hypertonic or hypotonic, but you see here for hypotonic we never want to give that to a patient because we do not want their cells, like red and white blood cells, to be destroyed. Isotonic is pretty much always safe. So here is a summary of the passive processes. We've looked at 3 processes that do not need energy. The energy source is just the movement of the molecules, so it's kinetic energy, but not active ATP use. The processes were simple diffusion, facilitated diffusion, and osmosis. Simple diffusion did not use proteins. Things were lipid soluble and just went through the membrane, or they were small and just went through the membrane. Facilitated diffusion used things like channels or carrier proteins to get things that are hydrophilic or large or charged through the membrane. And osmosis moved water through the membrane, and oftentimes channel proteins called aquaporins were used for lots of movement of water for osmosis. We had the movement of water in osmosis, but solute in simple diffusion and facilitated diffusion. So make sure that you know the differences. If you want to keep talking about solute through all 3 of these processes, know that water moves towards the areas of high solute concentration. And next for part B, we're going to be looking at active transport. Active transport does require energy. It's going to require ATP, which is molecular energy, and instead of moving things from high to low concentrations, we're going to see that with the use of ATP, we can move things from low to high concentrations, areas where there's not as much to areas where there's already a lot. So things that are too large for channel proteins that can't dissolve in a lipid bilayer, so even large hydrophilic molecules, or just things that are unable to move down their concentration gradient, can use active transport. An example would be amino acids and ions, and we're going to talk also a lot in depth about the sodium-potassium pump. So make sure to look out and watch Chapter 3, Part B as well.